So the webinar's being recorded. Everybody who has registered will receive an email with a link to the recording and also the slides. So don't worry about copying down links. You will have access to the slides at a later date. I am just going to quickly put a link in the chat though for everybody. If you haven't made an account, if you go to that page, you would be able to sign up for an account on the top right. Other housekeeping points, closed captioning is enabled. So if you want to use the captions, you can do that. There will be time for Q&A at the end. There is a Q&A box in the Zoom, so you should be able to add questions there. Not sure I can see them right now, but I have got Julia Kurtz, who is a new addition to the protocols.io team, and she is able to see the questions. So I feel confident that between the two of us, if you pop something in, we'll be able to see them and respond to them. If you do add them in during the webinar, if I do see them either in the Q&A or the chat, then I'll try and answer them if they're relevant then and there. It only works if I spot them. If not, we should have time at the end. There are other webinars, as I mentioned. You could sign up for an intro one if you haven't. There's one also about credit and publishing that we give regularly. And there is another one that I think in the new year we'll be doing, which is kind of slightly further on than this one, additional tips and tricks about some other new functionality we added this year. Okay, the agenda that we've got here, excuse me, I've got a frog. It's quite ambitious, so I will be going at a bit of a pace. So I hope everybody follows along, but it will be recorded. So if there's something functionality-wise that you want to replicate later, you will be able to come back and have a look at the recording of the webinar for that specific part. So I'm going to go give a little intro, a bit of information about using the editor, quite a few ways of getting your protocols in, what you can do when you're in there, how you can enhance them beyond something that's just narrative text in a materials and methods of a paper by using and including additional media like movies, images, tables, etc., and the components. Do a very quick view of the run and how you can keep run records, go through what exactly fork and copy are, and also versions, actually a little bit about creating protocol collections, how you can do that, what the value is there, and then sharing and publishing protocols. So we'll start with the intro. So this is just kind of step back. Protocols.io, if you're using it, is pretty powerful as a tool that can be used throughout the workflow process. So many people know of it as an open access repository, and that it's the place where you can post your final protocols. What people perhaps haven't realized that actually it's incredibly useful throughout the research process. When you start defining a new project and kind of think about background, what's been done before, are there any methods that are relevant? Quite often there will be publicly available methods on protocols.io belonging to someone else, maybe belonging to your own lab if you've been using it for a while. These protocols can be used and built upon. So then as you think about planning your approach, you can make your own versions of those protocols, build up a library of the equipment and materials that maybe you will use for that. And then you can start performing the research on protocols.io. I mentioned the run. This is what you can use to kind of keep trying new tweaks and optimizations to a method as you go. So while you're trying to really get it to work the best way possible, you can perform the runs on protocols.io, each time maybe making tiny changes and tracking what they are. The run records that you store will allow you to correlate what you did on a certain day with the data that you generate on that day, which obviously this might be an iteration you make multiple times until you successfully have a robust working, repeatable, replicable protocol, hopefully. So you might end up at success, hopefully, eventually you end up at success. And the experiments should be working. At that point, you can publish post publicly the final protocol. So if you're submitting a research paper, you have basically written the comprehensive materials and methods for that process and protocol, one or more perhaps, that will go with that paper, and you can make them public alongside the paper. And we have a way of keeping it private until a paper publishes as well. So we very much work alongside publication process for journals. Why would you do this? It is pretty certain that your most likely future collaborator is yourself. If you're not documenting and storing the information that you're doing, you will not remember. It's like when you tweak a recipe at home and something works really well, but you didn't write down what you did. You will not remember probably if you don't do good documentation using a tool like protocols.io. To be fair, you don't have to use protocols.io, do it however you like, but it is an important part of knowing what you did, tracking, and this is helpful not only as a public service if you make them publicly available in an open research friendly manner, but also for you in the future, for your collaborators in the future, and everyone else that you maybe work with in your lab or in your team. It's really important to include information to reproduce. So do unto others as you do unto yourself. The reasons being that there was a survey in Nature. It was a while ago, but I don't imagine actually that these numbers have changed hugely. 70% of over 1,500 scientists who were asked said they'd failed to reproduce experiments. This is a big problem because it's time and money expensive, and most research is being publicly funded or charity funded. It's a big problem if people are doing things in a bubble, not properly documenting, and then no one else can make use of the research that they're performing and build upon the approaches that they've come up with. This is just a nice example of someone who was really excited to start using protocols.io, the Chattiverdi lab. I think she was UC Riverside, I forget, one of the University of California campuses, who was setting up her new lab. And before her very first lab meeting, she had created a whole workspace on protocols.io with subfolders for all of the protocols that that lab is using, that she knows her team will be using regularly. So it's a great way if you had the opportunity to do this early on. It's Riverside, obviously, I've got it written there. If you have the opportunity to do this early on, as you set up a lab or as you start a new project, whatever the thing, if you create the order and then use it, it's easier than doing it post hoc and having to come back. Okay, I'm going to go into using the editor. I've got a few more slides to just go through how we have an import service you can use, and then I will go very much into demoing the platform. So we have an import service, which may not be known by everybody. You can get to this very easily if you're logged into protocols.io. If you click your avatar or icon that's in the top right and navigate to your profile, you'll see on your profile page that there's actually an entry for protocol entry. And when you click that, it brings you to the import service page. You can actually go directly to this page with the URL that's at the bottom here of the page. We enter protocols, which is named accordingly. You'll land here. This is a place where if you already have protocols in PDF or docx or whatever format you have them in, maybe they're in a Google Doc with a link, a share link, you can give us the file, either uploading a specific file or a link if it's something that's on a SharePoint somewhere or in a Google Drive. And then we have editors who have science backgrounds who will enter the protocol for you. Right now it's a manual process, but we are developing really good importers actually that are working incredibly well, either direct pass or GPT aided if it's more complicated. And they can get the protocols onto the platform for you. You then review, and when you're happy, we transfer it back to you. You can either keep them private or you can publish them at that point in time. So once, excuse me, once you've submitted the protocol, you'll get emailed status updates, but also from that protocol entry on your profile page, you will always be able to track the progress of any of the protocols you submitted. So you'll go here and down below. If you've got multiple orders, you'll be able to toggle between them and see what stage it's at. When you're reviewing your protocol, it's very easy. You use the commenting functionality. So if you want, you get the opportunity to ask the editors to make a few changes if something's not right. You would just add comments on the specific step where you want them to do something different. So it's very easy to do that. And when we're happy, there's a big approve button at the top, and then we would transfer back to you. So if using protocols.io feels like an effort, that using that service can be a big way to overcome the activation barrier, the activation energy needed to start using a new tool, because we can bring something in that you're already familiar with, and you'll immediately see how your methods can be represented on the platform, and it will use all the functionality in there for you straight away. Okay, now I'm going to show excuse me, I really have got a frog in my throat this morning. I'm going to show you how another way that's a bit of a cheat to put a protocol onto the platform. Although I'm not really, I don't know why I'm calling it a cheat. It's just functionality we've created to make it easier for you. So this is a completely brand new protocol that I've made, and it's going to be an example of how you can insert steps from text. So if you have a doc file and they're all in there, so I've got an example here. It's let me make that a bit bigger. A Google Doc that's got 16 steps in a protocol, a bit of additional information here. What I can do is copy the steps that I've got in this file. So if I copy those and then go back to the protocol, this new protocol that I have made, if you look over on the left-hand side, this is the toolbar for doing things with the steps and the sections. We have insert steps from text. So when I click that, hopefully it works. Of course, this is the problem with the live demo. Let me just reload that and see if it works. If I reload, prepped everything up earlier. This is not good. We'll try this one instead. No, I don't want to do that. Delete those. I am not sure why that's not working. Yeah, no, it's not insert from clipboard. Clipboard is when I've saved somewhere else. I think it's just a bug. I'm going to report it to my developers right now in hopes someone can fix it. But what I will do is I have a video of this when it does work, and I will put that into the slides because that is a complete fail in terms of a demo. What you can do with it is paste in the steps, and you can say each line is a new step or each number is a new step, and it will pull them all into the steps for you. Honestly, it works really, really well. That's not a good start to the Monday webinar. Let me carry on regardless. The other thing you can do if you're making a new protocol, obviously, you're working in workspaces. You can have lots of different workspaces on the left-hand side. If you've just made one for a new collaboration or a new team and you don't have time to make all of the protocols straight away, what you can do is just drag in all the files into this space. So you can just do upload files and put everything into this common space where you and your team members have access. If you're making new protocols, collection, things like this down here, you can do this all on from the new button that's right there. I'm going to go on to components instead. Sorry, I was going to try and just quickly report this in our Slack. Sorry, yep. Okay, that's in the issues live. That's not how it should work, right? So I'm going to go through components. Hopefully you've made a protocol or at least have played around. If you haven't, you'll know that you can see that it's a step-by-step format. And over here on the right-hand side, I have other functionalities that I've got, and I've picked here components. We sort of have templates. They're not really templates, actually. They're just preloaded sets of potentially the components you might need for biology, for a computational pipeline or workflow, or for chemistry. I've just got all here right now, and I'm going to run through some example things. I've sort of set myself prompts to illustrate various different things. If you have at the start of the protocol, you need to make the buffer, or there's some other kind of sub-protocol task that needs to be performed at the very beginning, what you can do is search for a specific protocol that you need to import. So if you've got one that's making a buffer, we don't have that here, but what's included in the PDF of your protocol, this is the EG of what to do if you're using our import process. you can put this sub protocol in, and then as a part of this protocol, you'll know that you need to be running this through. You can see what they all are at a glance before you can continue on. Nice and easy. We have volume, quantities, amounts, temperatures, all sorts of different things. So if I want to use the amount one, I will just be popping in the quantity. I can change the units really easily. I can add additional information if I want, and then I've got it nicely formatted. The benefit of using quantity like this, I'll show you off in a bit with the run, is that if you're doing a dynamic run, you can scale quantities. So if you are doing a master mix or making buffer and you want to generate more that day, you can very easily scale everything either in the whole protocol or in a single step. Sample is another nice component. So if you're kind of creating a specific sample that will recur throughout the protocol, what you can do is add sample. So I'm using the sample one over here. We'll call it my special mix. And I'm going to say this is created in, oh, my typing is appalling today, in step 3. And so then I have defined this, this entity, this sample specifically here. And then later on in the next step, for example, 5 ml of, I can just say sample and it will add in what it was that I defined. And then later on, it will, if I forget when I get to step 20, what is it? It will remind me this is what you've created in step 3. So it's a way of defining a specific sample through the protocol. We have, just to show if you've got a quantity, so if you do successfully get the protocol imported, pasting in from a file, what will not be there is the formatting of the protocol itself with components. So you'll have quantities in there. You can quite easily turn them into amounts by clicking on, like selecting the quantity and clicking on what it is. It's not going to put that reagent A in, but I could add that in if I wanted. But so once you use that pasting in functionality, you will have to still add the components and things here. Next one, we can add these kind of critical steps, these nice images to show what it is that you're doing at a specific step in the protocol. Very easy to put these in as well. So over on the left, we've got these 6 dots and mark as a critical step. So you can see I've got lots of nice icons to show what it is, what it is that I'm doing at that step. So if we're pipetting, we can do this. If we're mixing, we can add mix as well. So we can add a few different things together, and they show up really nicely. So then if you're viewing the protocol, those appear. So you can at a glance see a bit more easily what it is that you're doing at that step. So there are other things in there too, like this screen is moving around. Overnight step or a pause point, critical step. So you can really make it clear exactly what the things are that are happening at that point in time in a more visual way without having to look. So you can very easily see, in 3 steps, there's a pause point or whatever it is. We have duration as well, which is very nice. So I've got 30 minutes. If I click on duration, it knows that's 30 minutes, it's there, but I could type in the times here like this. I also could say overnight night. And I'm also getting a duration prompt. Do you want to add and update the total time to include this duration? So I'm going to say yes here. So what that is doing is adding a time for this particular step, how long this step is going to take. I've got another one there, 24 hours. We'll do the same thing. So that actually is going in as 1 day. So it's going to calculate from 24 hours and turn it into 1 day. And again, additional info or overnight and the duration. So we'll say yes. So we've got 1 day and 30 minutes. So that will also be adding to the duration of the entire step at the top. Section at the top. We can pop in notes. So you can add something specific, whatever's relevant. You can have it to show as collapsed as default, which means it will be closed when someone views the protocol. I'll illustrate that in a moment. We also have safety information. It's very similar. But I'll leave that one not collapsed. When I'm viewing the protocol, we'll do a quick toggle between. Tempted to rate us poor this morning after my bad experience there. You can see those icons, how they show up. This one I said load collapsed, but I can expand it very easily. This one, important info, I've left that open. So you can see what that does just from a visual way when you're actually in the protocol looking at it rather than editing it. Go back down. We have, these are really nice and I think really useful, but it's not the most intuitive to use. This is called Smart Component, and it allows you to include a key value table, which again, I think the terminology is not very user friendly. Basically what it is, it allows you to add, specify something and say if it is required. So maybe we won't require observation, but we will require number of cells. So if you're performing the protocol in the system, it will require an answer to that as a part of the protocol run. So you won't be able to move past until you've completed it. You know, like any special observation, maybe that one we don't require, but you need to be giving me a number of cells or something like that for this next part. You can have as many rows to this as you want. So it allows you to have a prompt for specific information that needs to be included during that protocol. I've got myself directive to mark this as a pause point. So we can see the nice pause button shows up on the right-hand side. Over on the left, I mentioned this is where you can do formatting things for the steps and so on. So we have step 9, but what we can do is add sub-steps. So if step 9 has separate parts, 1 and 2, we can add them in so they come up this way. And then you may have noticed I've got section 1 was purple. Actually, the color difference is not great. Section 2 is blue. I can insert a new section. And if I give that, we'll call it section 1.5, you can see that switched the coloring around. And what these sections are doing is creating a table of contents for me. That looks complicated. You'll see what those step cases are in a moment, but also that table of contents allows me to navigate. So I can jump up, I can jump back. So if you're putting together a complex protocol, you can have a nice section for the different parts of the protocol or different days, or however makes sense to separate up the protocol. Maybe you're performing it with a colleague. You can do that with the runs where one person does section 1, another person's doing a different section. That can all happen using the sections we've got here. If you were doing a methods section in a paper, generally you can't put much by way of imagery in there or dynamic kind of media. What you can do on protocols.io is include images. So I've just got a demo ping here. Similarly, using that upload image, you can upload small movie files from your phone if you've got them. They can't be too big to do them this way where it's directly importing into the protocol. You can give it a legend and you can resize. So you can just illustrate the protocol. Maybe it's a photo of the setup, maybe it's a photo of kind of expected results or what a sample should look like at this point. Maybe it's a little movie of how you're actually performing that step. You can do that doing this here. If it's a larger movie, I can't remember what the max size is for the video that you can import using that insert direct image or video, but it is limited. If it's something much bigger that's more of a how-to guide, you can use embed media. And so what you do there is you can have the video that you want to share on YouTube, on Vimeo, somewhere like that, and you can then embed the code which will pull that directly in. So I have a YouTube video. It's kind of genetic engineering how to, probably wouldn't be doing something like this, but it just to show you the example. On the YouTube site, you'll click share and embed, and we will copy the embed code that we've got here, and paste that into that embed media, and it pulls it straight into the protocol in that step. So when someone's running the protocol, they'll be able to watch the movie or not, depending if they've already seen it, they don't have to watch it again, but it's in there. So if this is instructional for someone new joining the lab on how to do something, you can really give them very detailed information. Okay, keeping an eye on time. Adding a table. So at the top, there's another toolbar. We have several toolbars. This one is more standard formatting type functionality. So we can add a table. And if we can change, I think we can change we can rename things. Oops, I'm not in there. Observation 1. So we can change these. Don't know why I've put a 1 in there. And similarly on the left. And also what we can do is put in Excel formulas into specific cells in here. And if then I actually run the protocol in a minute, I'll show you how that works. I'll pull that down. Works like Excel. You can drag the things across, so it'll do you the similar thing, but extend it. So that one will be a 3C3 and so on. Gives you the right, I know what I've done wrong. I needed to put that into the second one down. Like that. So this one should be A1 and C1. You'll see in the run in a minute, we can do a legend as well, and so on, formatting nicely. In the run, if I complete the table, it will calculate the thing on the fly for me. Step cases. So step cases allow you to take different pathways. So if you have got a different route for, maybe it's doing something at -80 or a different temperature or with a different sample at this point in time, what you can do is create step cases. So you'll be doing that over on the left-hand side, add step case. I can't do that. If I did this here, it would let me show you that one. Add step case. I already made one in this instance and set up first case. So first case will follow a different pathway. And again, then I've got another step case following condition A, condition B. Here I can add a second step case if I want to, a new case within this one. So we'll call this second case, case 2. Add a description. You can put a file if there's something relevant to the step case. And then what you see is that I've got a completely different pathway. There's nothing following. So then I can add new steps in here. And so on. And then that is this pathway will follow down whatever gets added under this case 2. Case 1 has a different pathway. What you can't do is merge them back to one another. So you can only, once you've diverged and made a bifurcation like that, you have to then follow going through. If subsequently you wanted to copy several of the, copy those steps and paste them into a different step case, I should be able to, oh, now that's working, insert from steps, and they'll get added to the end of this one. So I think I can go, I think the developers have fixed that for me. I'll go back and show you in a minute the insert scenario. So we made a step case, we can make another step case really easily. It's just going to ask me for the info. and it will add a step case. So I can add another one in here. So we can do as many as we want. So you can keep going as much as you want. Very easy to delete as well if you want to. Oh, I think I didn't want to completely delete that. 1 second. You can go back to, or not. Oh, I think I've done most of it. But the next part that was on here was to make a new section, I think. And I forget, but they've gone. But I think I was more or less at the end of what was in the step case part. What else was in the tables and formulas, components, attachments? The one other thing to show is that within a step, you can also add an attachment. So we have attach a file. So individually, you can just select a file. So if in a step there's some important information that comes in, you can either have files that you've already got in the file manager, or you can pick from your computer wherever you are. So you can add information that way. You also, under one of the other tabs, description, I think it is, can add a bigger attachment if you need to, and that would come maybe at the very top. That's the other thing I was going to show, was the plate map. So now it will show up at the very top of the protocol. So if there's something you want a user to see right at the start, that will give you that plate map attachment at the top. So if you want it to be something that's seen very prominently at the beginning, you put it at the start of the protocol. If you want it to show in a step, you can put it in a step that way. So let me just go back to edit. So the last component I'll show is the plate maps. There are more in here. There are things like you can define code or link to software, all sorts of different things, databases. It's hard to show everything in one thing, but we've got preloaded well plate maps. So if you're doing any kind of screens, you can do this here. So you can define rows and columns or put your own custom numbers in. When you've put them in, there are some template color settings. You can pick the colors, save. You can do each one individually if you want to edit an individual cell color. And if you type something here, A1, it will give labels to the cells. So you can see kind of how that works. Copy a whole thing. So if you've got multiple and you want to copy paste it, you can copy and paste the whole well plate map. There's additional functionality that we go over in the more detailed one, which is how you can import in a file and have it kind of pre-populate. It won't bring in colors, but it will bring in the contents of the well plate map if you set it up correctly, but it's a little bit complicated, so I won't go into that now. I see a question about, will there be a database of the reagents you entered into the protocol? So what you can do in a workspace is manage reagents and create a library of reagents in there. So you can see I've got a few in this workspace that I've defined. I can search. We pull in a lot of things from, don't know why that's not, here we go. From catalog vendor catalogs. So we can pull in a lot of different things. We can add that in and put all of this information into just a library catalog just for this workspace. Sorry, I'm struggling to find my words. Make a library for that workspace. But when you're in a protocol, sorry, I'm trying to hide trying to get to the tabs without the zoom thing there. When you're in the protocol itself, it's just a step where we are. Here we are. So we can actually go to the reagents. No, I can't. And equipment works exactly the same way. If I could see the reagent one, there it is. You can look just at your workspace reagents. or you can search everything, or you can create a new reagent. So you can then pull in specifically, but then, or if you've made a kind of library for the workspace, then anyone in that workspace will be able to search just from the reagents that have been put into the catalog, the library that you've made for that workspace. But there's a ton of other stuff in there, so we've sort of left it so you can either search everything or just the workspace reagents, and make new if you want to, add other things that aren't in there, as whatever makes sense to you. So there's lots in there, there's a lot of functionality. Right, I'm very quickly going to go back here now and see if this works. I'm kind of feeling hopeful that it will do. So we'll do import steps from text. No, it's not liking it at all. Let me try a reload. I did get an answer in the chat, but I don't know what he said. Oh, maybe not. It seemed to work before when I when I'm going to do a very quick, oh no, I'm not in the file manager now. Make a new protocol and see if it works. I don't want to spend more time on it because it might throw me off. There's a few more things to show. I don't know. I thought it came up earlier when I clicked when I was in here. That was the annoying thing. Never mind. We'll carry on. The run record is the next thing I will show very quickly. If you've got your protocol or someone else's protocol, one of the public ones maybe, what you can do is a run of a protocol, and you'll say where you want to save the run record, and then you'll start the run. It will open up functionality to be able to step through all of the steps that are in that protocol. You can define an experiment ID if you want. So if you're tracking in some other way the ID versus what you're going to do that day. And then what you can do is step through. So that very first protocol that I pulled in, it's actually giving me all of the steps in that protocol. So if I say done, it will actually tick all of them off in one go, but you can step through them individually as well. We've got a component step, we'll click that one, done. This one that's got a few different quantities in, what I will do is I can edit or scale. If I click edit and scale and then settings, you can see I've got the option to scale the step. So if I want, I can scale up all of the items in that step, and then it will be giving me just exactly what it is, and it tells me each time scaled by 5, and we can save that here. So we'll step through. I can, excuse me, I can also add a comment. So if something happened here or an observation, I can add a specific comment, and it will get stored at that point on the protocol as well. When I have a timer, I can actually run the timer in here, and it will sound a little alarm when it's finished. So if you're doing something and you want to kind of track it, you can do all of that during the run of the protocol. And what that has done for me, where am I, the file manager, is in my run records, that's this component examples, November 18th, that's the one I've just done here. And it's grey because it's not been completed. It tells me it's incomplete. These green ones are complete. So if I'm stopping at lunch, I can stop, I can turn it all off if I want, and then come back and resume, and I can click to resume that run record. Or another team member in this workspace could also pick it up and finish it. So if it's over multiple days, different people involved, you can start this run record as you like. Different parts could even be being done by different people at the same time, if you've got sections that can happen synchronously like that. It's very nice functionality. Okay, next thing I am going to talk about is fork versus run. Sorry, fork versus copy. So if I've looked online and found a really neat protocol, someone else's, that I would like to make a version of for myself, what I can do is make a fork. So you can see actually I forked this component examples from another one called exactly the same thing. So I try and do this so that I have a new clean version of the protocol for each time I run this webinar. I can just make a new fork and it will let me complete everything again. That's maybe not the most standard way that you would use it, But let me give you examples of those different functionalities we have. So very quickly in a slide, just to talk these through. So for a protocol, either published or not published, what you can do is make a new version. If we assume this is on a public protocol, you can use this. These are use cases for each of the different scenarios. You can correct an error, or you might want to add more information about optimizations. Maybe you've got new something new that you know just is working slightly better. You can put this all on. Maybe you've got a slightly new piece of equipment and you just want to show you how to optimize further using that. That can then be made and published as a new version. So you can add a note when you do that about what's new. And those public versions are always linked so that each one will have its own DOI and you can track back between the different versions. All users can make a new version of a protocol, but important to know, only the owner can publish it. If you were trying to make a different kind of adaptation of someone else's protocol that you want to publish, the way to do that is with a fork, not a version. The version is really for the same user to be making updates. However, if it's owned by a group leader and they want the student to make the updates in a new version, then the student can make a new version. The owner would have to allow it to be merged back and can then publish it. Fork allows you to create a carbon copy of anyone's protocol and it will track back that lineage. So this might be if there are slight adaptations as a use case. So maybe you have completely different equipment or some other setup in your lab that you're going to need to implement. Maybe you've got just slight modifications for different species, different sample, different cell type. And then you can have, you know, this is protocol A for kidney cells. This is a fork of that, and I've had to make adaptations to run it on liver cells, for example. And that fork will be visible like the one I just showed you on my component examples. Copy is different again. Oh, I should say fork, obviously available to everybody. Publicly, people will access any public content and can make forks of them. Copy is different. You can only the owner or the co-admin of a protocol, so someone who has kind of oversight of a protocol, is able to make a copy. When you might want to do this is if you've got a kind of template in your own lab where you know a certain amount of the information will be the same. So perhaps you've got same abstract, same authors, affiliations, acknowledgements, grant funding information. You can make a copy and all of that info will be pulled across. Maybe you've got same reagents or things that you're using as well, and so on. There is a more detailed help page on fork version copy, which can be quite useful to people. I have got, before I do Q&A, a couple of quick other things to show. One thing to say is that you can make collections of protocols. So I've shown a lot about an individual protocol, but it's very easy to make a collection as well. And the scenario when you might want a collection is best exemplified here by one that's made public, where there are protocols that all correspond to one paper. So these authors have pulled in protocol 1, 2, 3, 4, 5, 6 different protocols as a part of a paper that was published on eLife. And we can always add the link to a paper. So if you're doing this alongside a paper, we can put that together. Each protocol in there has its own DOI, but the collection itself has a single DOI. So what you can then do is point to a collection of protocols on protocols.io in the body of the materials or methods of that paper, and anyone who clicks will be brought through here. I should say, if you come from the paper and it's been updated, which this one I happen to know has, you would land on this, which gives you an alert that there's a newer version of this collection available. We call this dynamic permanence. This means that authors can make new versions. The paper will always link to the version that was published at the time the paper was published, but authors can come in and make new versions. So we'll say close, but I can see here, actually this got updated in February 2024. So the authors might have made some changes. When changes are made, one sec, let me go to version 2. When changes are made, Well, we should be able to make a comparison, but maybe if I opened one of those that's in a second version. Oh, I don't know what it is. You should be able to track the changes from one version to the next when you've got multiple versions of a protocol this way. And to pull protocols, if I clicked make that new collection earlier, this is what it looks like. It has the same space for abstract, author information, everything like this. But then within the collection itself, what you can do is search for all of your protocols. So maybe we'll pop in poster draft, sticky toffee banana bread, obviously really important protocols that I've added. And they all get added in a link. It's really easy to say that's number one, that's number two, to move them around in here. And when you view, they're all in there. So you can then have that collection of protocols really very easily. The last couple of things to show is share and publish. So if I'm on my, well, let's go back to edit. If I'm in my collection, or if I'm in the file manager, I've got a very prominent place to share. By default, anyone who belongs to this workspace that I've made this in will have access to this protocol. But if I want, I can share specifically with another person. So Lenny, my boss, isn't in my workspace. So I can add him, but like this. I think he did show up. I'm in the wrong one. I can put Lenny in protocols, add him here, give him access, but I also can control the permissions that he has. So maybe I only want him to be able to view it. I don't want him to necessarily edit or publish, or I can give him those rights if I want. Just need to save. Also, maybe I'm working in my workspace, but I perhaps want to share it afterwards with a lab workspace. I can say I want to share with a specific, maybe I want to put it in the +1 lab protocols workspace. I can add that here. And again, I can control the permissions that are in that workspace. So I can do lots of different ways of sharing with other people to get feedback on a protocol. And the last thing is publish. If you've made one protocol, or if you've made a collection of protocols, you should hopefully see a very prominent publish button at the top left. We always advise using publish straight away. If you don't want it to be publicly available until the protocol, a corresponding research paper is published, you can use reserve DOI. That will tell you what the DOI will be, but it will also give you a private link so that you can give reviewers and editors access to a private link. But if you click publish, what's nice for a collection is that it will, if you haven't published the sub protocols already, it will publish them all at the same time. So you can put the whole thing together and save yourself quite a few steps. I think the ones I put into this collection were all already published, so it only shows me the one for the top level protocol. You will be made to do something, so it won't let me continue until I add an abstract. I don't need to add a disclaimer notice, but good to know maybe if you should. You're going to be made to include either a grant funder info or say doesn't apply. You're going to be made to add an author, minimally yourself generally, but you can add other people and so on. We've got some author guidelines. If you've used commenting functionality, you can strip them all off, go through all the other steps, status, protocol type. Do you want it to show up in any workspaces or not? And there's just a nice little preview option. And right at the end, if you click all the confirmations, if you've got a paid premium license, which you might have through an organization, once you've clicked those three and publish, it will immediately be published. If you haven't got a license or a premium account, it will enter a screening process. And so we screen everything just in case spammers do try and put content on the site. But it only normally takes a day or two. But if that just so people are aware that that might happen if you don't have premium access. But once you click, that's it. It's done. It has a DOI. It is permanently available, and much like a paper, you know, formally you should be confirming that your co-authors and supervisor, anyone on that or associated with that protocol, agrees with that. So I won't publish this one because it's not a specifically interesting protocol, but I'm going to stop there. I have run through a lot of things. Actually, let me put the page up that's got all the different links on it, and we'll go to Q&A. If anybody has any questions, or the webinars bit at the bottom is cutting off, but there's other resources there. That's just protocols.io/webinars. It's cutting off on my screen anyway. But I didn't see if any questions got put in apart from one earlier on. And I'm not sure if anything got added into the Q&A because I definitely can't open it. I don't know why today.
I've got the Q&A open, but no questions have come through just yet.
Well, thank you, Julia. Well, I'll sit for a couple of minutes because sometimes it can take time to type something in. So feel free to add questions. And if not, we'll be finishing up in a minute. I'll turn my camera off and leave you guys to add questions. I'll pop it back on if anything comes up. And I will stop the recording as well.